so you're not there. Fine, never mind. Okay, so I suppose we completed talking about ELISA techniques in the last class. And we spoke about different types of ELISA techniques. That is, we talked about the indirect technique, then we talked about the sandwich technique, and then we talked about the competitive ELISA technique. If you're having any doubts in those ELISA techniques, you can just get back to me once again. So having completed those, we will talk about the next very important technique, that is the polymerase chain reaction. That is the most important one over there. You might be asked this for the examination point of view also, polymerase chain reaction. So what is exactly polymerase chain reaction? So this is the instrument that is going to be used for polymerase chain reactions, and these are the test append-off tubes that you'll be going to be using for this chain reaction. That is, you're going to take a small segment of the DNA and you're going to get n number of copies of DNA, because you're going to amplify the DNA segment that you want to check out for any. It can be used for either knowing what type of a disease a person is affected with, or you can also use them for forensic science, therapeutics, if you want to get to know some proteins in them. And there are various applications for this polymerase chain reactions. This is what you're going to talk about. If you are going to have any doubt in between, you can just stop me and ask the questions over there and get your doubts clear. Okay, so we'll talk about polymerase chain reaction. So what is exactly polymerase, which is also abbreviated as PCR? We have heard a lot of this PCR technique during COVID also, because we wanted to know a person affected with COVID, what type of a virus it is and all those stuff over there. So it's a laboratory technique for rapidly producing or amplifying millions and millions of copies of a specific segment of the DNA. So if this is a specific segment of the DNA, one segment of the DNA, you're going to amplify it into n number of copies, as much as you want, which can then be studied in greater details. So what is the steps? The step is going to be 2 to 3 steps over there. So first thing is, step 1 is denaturing, step 2 is annealing or renaturing, and step 3 is elongation. So how is all this done is what we're going to talk about in this particular polymerase chain reaction technique over there. So when you're going to talk about aquatic biotechnology, how is this going to be useful is what we are going to study over here. That is, various molecular biology techniques continue to show importance in the fish and shrimp farming, particularly in detection and prevention of various diseases. So when we are talking about aquatic biotechnology here, we are going to give a lot of importance to aquatic culture, that is freshwater fish culture, because if you're going to talk about those that are going to be cultured at a Deccan Plateau or something place over there, where you don't have any sea waters over there. So here you're going to talk about freshwater aquaculture. And also we must have heard of this blue revolution also, wherein the central government is giving a lot of importance to the blue revolution, as people are turning towards fish eating population, because they don't want to have red meats, because to avoid any cholesterol. Red meat gives a lot of cholesterol. So in that case, you can shift to fish eating population. Why? Fish has a lot of proteins, and in the at the same time, it is going to have less of fat or lipids in them. So when you're talking about this aquaculture, you have to give a lot of importance for its health purpose also, because if you're going to grow them in an overcrowded condition, what happens is they might also have some diseases. So you have to detect them and prevent those various diseases, so that you can have a proper culture of the freshwater fishes. So one of the most important technique over here we'll be talking about is PCR after ELISA technique, and this has a greater impact and on the practice of the molecular biology, wherein you're going to use this PCR. So this PCR amplification of specific nucleic acid sequence was introduced by there were different researchers, but Psyche et al. 1983. They had introduced the specific amplification process, and has subsequently proved to be one of the most important scientific innovations of the past decade. With this technique, one can rapidly detect a virus or a bacteria or few copies of mRNA, because you want to know if a fish is affected with some diseases, you want to know what type of a disease that has been affected, whether it is a virus, if it is a virus, what type of a virus, or if it is a bacteria, what type of a bacteria. You could also amplify a few copies of mRNAs. You can rapidly synthesize them, clone them, sequence them virtually, and segment of DNAs can also be done in this particular technique. So conventional PCRs is a technique used to amplify trace amounts of DNA and RNA from a sample. So it can be even trace very, very few amount. Say, for example, when you're going to collect some information or proof from a place where a crime has occurred, one hair strand is enough, or a few, if there's any semen samples, you can use that also, or you can also use a trace clotted blood stain also is enough. You can just take those trace amount and you can just amplify that particular DNA or an RNA from that specific site. So a thermal cycler, what do you mean by a thermal cycler? The cycler which is going to give a lot of temperature. So temperature maintained, you can either increase the temperature or decrease the temperature. So thermal cycler is used to produce large amounts required for the research. And PCR makes billions of copies of specific DNA fragment or a gene, which allows detection and identification of the gene sequence using visual techniques based on size and the charges. So this can be used for a wide variety of laboratory and clinical applications, and forensic labs use it to analyze DNA samples from a crime scene. So that's what I was talking about over here. And this is also used to diagnose patients infected with any type of virus. This is also useful for pharmaceutical research to analyze and duplicate DNA and RNA samples for the use in the manufacture of the drug and the vaccine. Why? Because if you know a particular protein which is going to be important to produce that particular drug or a vaccine, then you should know what it is characters. So this definitely is helpful for the pharmaceutical research. So if you can see in this picture over there, this is what we were doing in the COVID-19, where a swab from the nose was taken, and this was mixed with the lysine buffer over there. So that's lysine buffer over there. And then this was sent on the RT-PCR machine to tell whether the patient was suffering from COVID-19 or is it something else, because with this process, we will know what type of a virus is affected, which strain of that virus is affected, and all those over there. So that is where we use this RT-PCR. And this is also used, the PCR is based on using the ability of DNA polymerase. Polymerase is here, we are going to talk about an enzyme to synthesize new strands of DNA complementary to the offered strand template. So here, what you're going to see over here is when this segment of DNA is there, when they are separated, the DNA polymerase is going to make the copies over there. So they are going to help in making these copies. How they are going to make? Because of the help of the DNA polymerase, which is an enzyme. So because DNA polymerase can add new nucleotide only onto the pre-existing 3' OH strand, it needs a primer to which it can add the first nucleotide. What is the first nucleotide? Here we are going to talk about the nitrogenous base pairs over there, which is going to add to the growth of the strand over there. So this requirement makes it possible to delineate a specific region of template sequence that the researchers want to amplify. And at the end of the PCR reaction, the specific sequence will be accumulated into billions of copies like this. So we started off with a single strand over there, and we are going to end with billions of copies. So how was this done? So from one copy, you're going to make two, from 2 to 4, from 4 to 8, and so on and so forth. So how is this possible in a PCR technique? That is what is called as polymerase chain reactions. So it's a chain reaction, continuous reactions over there. So here we are going to talk about the principle. So what is the principle of PCR? It is based on the enzymatic replication of DNA molecule. What is the enzymatic replication of the enzyme? Is a polymerase enzyme, DNA polymerase enzyme. So this is based on the enzymatic replication of the DNA molecule, and it is used for amplification of the specific region of DNA strand. So you're going to amplify only a specific region of that DNA strand. That is, here it depends on what type of experiment you are going to do. So in most cases, polymerase chain reaction is used to amplify DNA fragments of anywhere between 0.1 kilo base pairs to 10 kilo base pairs. So it can be as small as 0.1 to as big as 10 kilo base pairs. But sometimes they are also seen that it can also go up till 40 kilo base pairs. According to the PCR principle, DNA amplification is carried out in 3 steps. So you have to remember this. You can just note down in your notes what is this 3 steps over there. The double-stranded DNA of interest is denatured to separate it into individual strand. So denaturing is you're going to separate the DNA strand over here. That is called denature. So first sequence is denaturing the DNA strand. So that is the first step. In the second step, each strand of the PCR step is then allowed to hybridize with a primer. So after separating these two, you're going to have 2 strands. Now each strand is going to get copied. So that becomes 2 strands separately. So that is called as hybridizing with a primer. The primer template duplex is used for DNA synthesis by the enzyme DNA polymerase. So that is what is enzymatic replication. So enzyme DNA polymerase is used over there. So these 3 steps, that is denaturation, renaturation, D and re. D is to separate, re is to join them. So denaturation, renaturation and synthesis or elongation are repeated again and again to get or to generate multiple copies. So you can get millions to billions of copies over here. So that is what is PCR technique. So we'll talk about it in detail over here. So what are they? So the essential requirements, you have to note down this also, because without this, you won't understand what we are talking about in PCR technique over there. So DNA template, so that's a target DNA. It can be any target of your interest. It can be anywhere between 100 to 35,000 base pairs in length. We were talking about kilo base pairs. Here we are just talking about base pairs. So when you're talking about base pair, it can go up to 100 to 35,000 base pairs in length. Oligonucleotide primers. So oligonucleotide primers is the most important one. So you need to have 2 primers. Synthetic oligonucleotide of 17 to 30 nucleotide length. Synthetic is man-made. So you're going to produce this oligonucleotide base pairs, which is going to be complementary to the region flanking the target DNA. So what it is when you're going to talk about a particular DNA strand of your interest, you know what is the complementary nucleotide base pairs they have. You're going to prepare primers, which is going to be complement to that particular place where they have separated. So that is where you're going to prepare 2 primers over there, which is going to be complementary to regions lacking the DNA strands over there. Then what's the next one you need? You need a deoxyribonucleotide triphosphates. So this is a 4-deoxyribonucleotide triphosphate. What are they? ATPs, CTPs, GTPs, and TTP. Adenine, cytosine, guanine, and thymine. Sorry. So when you're talking about these deoxyribonucleotides base pairs, this is the one that is going to help in growth of the DNA, because that is a nucleotide base pairs of your DNA. So you need all these 4 one. That is DATPs, DCTPs, DGTPs, and DTTPs. Then the next one, what you are going to need is DNA polymerase. That is the enzyme we are going to talk about. So what is this DNA polymerase? There are 3 or 4 different kinds of polymerase as of now. But what exactly here we are going to talk about is a TAC DNA polymerase. TAQ, TAC DNA polymerase. TAC is a thermophilic bacteria that is present in the springs or volcanic sites. Because these TAC DNA, you can find them present only in very hot places. That is hot springs, some places where the water is going to be boiling hot. Even if the water is going to be boiling hot at some places, that is springs or volcanic sites, you find that this bacteria is still surviving even at 95 degrees centigrade, they are still surviving. So that is why you're going to use it over there. So that is the importance of Taq DNA polymerase. That is, we are going to use this enzyme to elongate your DNA, even though you're going to give a temperature of 95 degrees centigrade in this polymerase chain reaction. So if you remember, when you're talking about other enzymes, these other enzymes are going to get degenerated because a little bit of heat, you know that proteins and enzymes can get degraded. So this particular bacteria, which is going to be present in these hot springs or volcanic sites, where you have this geothermal vents is where they have to burst open in the volcanic places. They found that this bacteria was able to survive over there even at the high temperature. So we are going to use that particular enzyme that is called the Taq DNA polymerase, which is bought from this thermophilic bacteria over there. So a DNA polymerase that can withstand a temperature of 95 degrees centigrade is what we need to elongate the DNA. Okay, then you need a buffer system, a buffer solution that contains magnesium ions or potassium ions, which provides a suitable chemical environment for DNA denaturation and renaturation. Then it is also helpful in polymerase activity and stability. So it needs a buffer system wherein it's going to contain magnesium and potassium ions, which is going to give a proper environment for the DNA to denature and renature. So that is what you're going to talk about. So you need a DNA template strand over here, and this is the 4 nucleotide base pairs, the guanine, adenine, and the thymine, and the cytosine over there, the purine and the pyrimidine deoxyribonucleotide base pairs. So these are the essential requirements. So you need a template, you need 2 primers, then you need 4 nucleotide base pairs, then you need a DNA polymerase, that's an enzyme polymerase. And then you need a buffer system, which is going to maintain or give a optimum environment for the process to occur. So technique. So we'll talk about what is the technique over here. The actual technique of the polymerase chain reaction involves repeated cycles by amplifying the target DNA. Commonly, the polymerase chain reaction is carried out in a thermal cycler. So this is a thermal cycler and a small reaction tubes. So you're going to add your DNAs in these small reaction tubes. So you can see how small they are. The amount varies between 0.2 to 0.5 ml. That is 200 microliters to 500 microliters volume is the amount that you can add in this. And these tubes goes into this PCR thermocyclers. This is a plate where it is going to have the temperature. And then you have those buttons over there wherein you're going to give all the instructions when it has to increase in temperature, when it has to decrease in temperature, and how many cycles it has to undergo. So all this is done in this particular PCR machine, which is also called as thermal cycler, and you need these tubes over there, along with the materials that we had discussed earlier. So what is this actual technique that is going to be involved in this particular cycles over here? So each cycle in the PCR technique has 3 steps, as we talked. They are denaturation, renaturation. Another name for renaturation is annealing. Annealing is coming close together, and elongation techniques. So what is this? All these are related to temperature and time. So all these 3 steps depends on time and temperature. And each cycle might take 3 to 5 minutes, depending on how much of DNA is present and how much of primers are present and how much of nucleotide base pairs are present over there. So there's an automatic machine where you can see on the y-axis you have the temperature, and on the x-axis you have the time over there. And when you increase the temperature at say around 95 degrees centigrade, you are allowing the DNA to denature. So that is what we are going to talk about. At 95 degrees temperature, you are the hydrogen bonds between the DNA breaks open, and then they are going to get separated. And then when you reduce the temperature, reduction of the temperature at say around 55 degrees centigrade, the DNA is going to anneal or renaturation is going to occur. Annealing or renaturation is going to occur. So that depends on what is the time you want to give. So here in this picture, they have given 1 minute. It can be 2 minutes. It can be 3 minutes, depending on your laboratory practice. Then you're going to again increase the temperature to say around 75 degrees over there. And when you're going to increase the temperature, DNA synthesis or elongation of the DNA strand is going to occur. That is, you had separated the DNA and then this 2 DNA has to copy and elongate. So that is called as DNA synthesis. From single DNA strand, you're going to prepare 2 DNA strands over there. So that is what is called as DNA synthesis or elongations. So this is the technique that we'll be talking about of the repeated. So we'll talk about this in detail one by one. So if you can check over there. So you're going to talk about denaturation. So what is denaturation? Before that, you can check this picture over there. So that is the DNA strand, the double coil strand over there. And this double coil strand will always be joined by the hydrogen bonds in between. So if you can see in this picture over there, so you have this hydrogen bonds over there, adenine to thymine with a double bond and guanine to cytosine with a triple bond. So this is the hydrogen link that is present between these 2 DNA strands over there. And when you increase the temperature, this hydrogen bond is going to break up and then they're going to be separated into 2 strands like this. So in between, you have this hydrogen bonds. So this hydrogen bond is going to break up. And as they break up, these 2 strands are going to get separated. That is called as denaturation. So when is this going to occur? When you're going to raise the temperature to 94 to 95 degrees centigrade for about, say, from 0.5 to 2 minutes or 3 minutes, depending on what is the type of DNA that you're going to use. The DNA is going to get denatured and the 2 strands are separated by breaking hydrogen bonds between the 2 strands of DNA. So this is what you're going to see over there. Now the single strand of the DNA is now going to act as a template for the production of new strands. So you have 2 templates over there because the single DNA that has broken over here. So you have 2 templates and these 2 templates acts as the template for synthesis of a new DNA strand to occur. So the temperature is the in the denaturation of the polymerase chain reaction should be provided for a longer time to ensure the separation of the 2 strands. So if the 2 strands are not going to get separated completely, then you may not be able to produce a new strand over there because the primers has to attach onto the opposite side. So what is this primer attaching onto the opposite side is what you're going to talk about in the second step. So I hope you have understood denaturation. Now we'll go to renaturation or annulate. Renaturation is again you're going to prepare join this DNA. How we are going to join this DNA? Now if you can see in this picture, you have the primers over here. The pink color ones are the primers, the oligonucleotide primers. These are the synthetic primers. Because why synthetic we are saying? They are going to join or they are going to attach onto the complementary site where they have to go and join, because they have to have a complementary nucleotide base pairs over there. Otherwise, you're not going to join over there. So these primers are prepared. Then you have the DNTPs. What are the DNTPs? All the 4 nucleotide base pairs you need. Then what is this brown color one? This is the polymerase, the Taq polymerase. From where did we take it out? We took it out from the Taq or the thermophilic bacteria over there. So that's called as Taq, T-A-Q. Taq polymerase is an enzyme from the bacteria out there. So what is happening? When you want to re-annual, so this is the denaturation, that is at 95 degrees. You have separated this DNA into 2 strands over there. Now this is the template strand over there. Now once this has been separated, you're going to decrease the temperature. So what is the temperature that is, it needs to be decreased at 55 degrees centigrade temperature. From 95 degrees, you have decreased it to 55 degrees centigrade. Now, when the temperature is reducing, what happens is the primers that are present in the solution, these are going to find out where those DNA complementary strands are present, and each one is going to attach onto the opposite side of the strands of this. So the copying occurs from 5' to 3'. Why? Because the nucleotide base pairs has to be added only at the 3' region. That is the reason why they are going to be copied from 5' to 3' regions over there. So they're going to attach, these primers are going to get attached onto the opposite sides. And once the attachment of the primer is done, then the elongation occurs. How your elongation is going to occur. Am I audible? Because my internet says that there is some problem. If there's any problem, please call me and ask me for any doubts over there. So if I'm audible, then I'll continue with the class. Shweta, am I audible?
Yes, ma'am.
Yeah, you can hear me? Right.
Yes, ma'am.
Okay, fine. Uh, yeah. So such a process is called renaturation. What is renaturation over here? We are going to find that the DNA is going to be combined over there. That's called as renaturation. And a high concentration of primer ensures, because you have to need a lot of primer over there. So high concentration of primer is also necessary over here. So high concentration of primer ensures annealing between the DNA, each DNA strands and the primer rather than the two strands. What it means over here, if you can understand this line over here. A high concentration of the primer ensures this annealing over there between each DNA strands and the primer, each DNA strand and the primer, rather than the two strands itself. So if you're going to have less number of primer, the denatured DNA strand that you have done, this is when you're going to decrease the temperature, these two again going to get re-annealed. You don't want that to happen, because these two strands are the templates, so that you are able to copy a new DNA strand on each side of it, like that, not like this. So when is this possible? When you're going to ensure enough primers that is to be present in the buffer solutions over there along with the DNA strands. So that is what the point you have to remember. So the two separated strands in the polymerase chain reaction run in the opposite direction. So this is the opposite direction. They are going to run from this end to this end, from that end to this end over there. And therefore, two primers such as forward primer and reverse primer is used in the renaturation. So that is renaturation or re-annealing that is done with the help of the primer. So that is the second one. So now that the primer has gone and attached over there, so that is the annealing. Now we'll talk about how the elongation occurs. So primers we spoke. Now we are going to talk about the elongation. So what is happening in the elongation over there? So in the elongation technique again, so what you're going to do is, so can you see the primers is attached and the Taq polymer is also is attached on to this point, and they are going to elongate over there. Now for this elongation, you're going to again raise the temperature from 72 to 80 degrees centigrade. So first it was 95 degrees, then you came to 55 degrees. Now you are going to again slowly raise the temperature to 72 to 80 degrees over there. And initiation of the DNA extension occurs at the 3' hydroxyl end of each primer. What it means over here? DNA extension, that is addition of a new nucleotide base pairs occurs at the 3' end of the hydroxyl end of each primer. So the primers are extended by joining the DNTP bases complementary to the DNA template in a 5' to 3' direction. So this is a 5' to 3' direction, and from there 5' to 3' direction. And where the nucleotide base pairs is added at the 3' OH region. So this is where the new DNA strand, the DNA base pairs. So all these colors over here are different base pairs complementary to the template strand over there. So each one has to be added to the growth or elongation of the DNA that is going to occur. And how is this done? This is done with the help of the DNA polymerase. So that is a Taq polymerase over there, the blue color one, that's a Taq polymerase, and this Taq polymerase are the one that is going to add on the nucleotide base pairs, and it is going to elongate over there. So the extension time depends on both the DNA polymerase used and on the length of the DNA fragment to be amplified. If the length is too small, the cycle is very fast, that is the elongation period is very fast. And if the DNA fragment is long enough, the time that it has to elongate will also take time. So it all depends on the size and the amount of DNA polymerase that is present. So the enzyme Taq polymerase adds DNA bases to the single strand. Therefore, a double strand DNA molecule is obtained during the elongation. So this is how the elongation occurs over there. So we talked about denaturation, we talked about renaturation, and we also talked about elongation. How is the elongation done? With the help of DNA Taq polymerase and the nucleotide base pairs. That is when the primer gets attached onto the separated DNA strands. First, the primer has to go and attach onto the DNA strand. And then from there, the Taq polymerase goes and attaches onto this primer. And then the elongation occurs. How the elongation occurs? By adding in the nucleotide base pairs, you're going to elongate the strands over there. So that is the elongation of DNA strands over there. So that was about the polymerase chain reaction. That's a important and compulsory question for your examination point of view, because they will ask you all what is this polymerase chain reaction. You're going to answer this question by giving appropriate diagrams over there. So once again, this is a very beautiful diagram I found out from the Google. So I just wanted to show you all how they work over there. So how it is done over there, you have a DNA sequence of your interest over there, and you're going to have a primer that is going to be specific to the strands that is present over here. Can you see the strand over here? This strand, light blue strands over there. So this is specific to that primer. And then you need a DNA polymerase, which is a DNA polymerase over here. Here I spoke about TAC polymerase, TAQ, TAC polymerase. Polymerase is an enzyme. Now, what is the first step over there? In the first step, you're going to denature the DNA strands. So this is the strand that you have denatured. Denature is we're going to separate them. And once it is separated, the first thing what happens is, once it is separated, the primers are going to get attached onto the strands over there on either side. So it is going to get attached over there, and they are going to get elongated from 5' to 3' directions over there. And once the primer has attached, immediately the TAC polymerase is going to attach onto this particular growth of the DNA. And once the TAC polymerase attaches, it is going to add the nucleotide base pairs, and they are going to elongate in this direction. That is the opposite direction. They are going to get elongated. So this is the first cycle that has occurred. The first cycle is over. Then when from one DNA strand, you have got 2 strands. Now this 2 strands is going to become 4 over there. So how is this done? That goes into the next cycle over there. So in the next cycle, what happens is, this new strands that have been prepared, now this undergoes again denaturation. Now this again gets separated into 2 over there, and again a primer is going to get added, and they're going to add new strands over there, and that is how you're going to get 4. And then this 4 again goes to the third cycle. Again a denaturation occurs, again a renaturation occurs, and again an elongation occurs, and thus this 4 becomes 8. And after say around 20 to 30 cycles, you can see millions of copies. So this particular single copy over there is going to be made into this millions of copies over there. You can see that many millions of copies is how you're going to prepare. That depends on how many cycles you have asked your PCR to run. It depends on that many cycles. If you're given only 10, then you'll be getting only few numbers of copies. But if you're going to make a 20 or a 30 cycles, then you're going to get millions of copies of that single strand. Now, why are we interested in preparing that many copies? There's an application for that. So you're going to prepare, for example, as I said, at a crime scene, you want to find out who is the culprit. So you might find some samples on the victim's body, say in the nails, or they must have pulled out the hair. The hair strand has those DNA bulbs over there in the hair, or maybe a blood stain, or anything could have happened when they're scratching. They must have scratched the skin. The skin will be lodged onto the nails over there, or the blood must have been, or a semen sample for that matter. All these can be collected because you're going to be very few DNA. You cannot be able to do the any type of test on the few strands over there. What you're going to do is, that amount that you have got, you are going to make the cycles, amplify the DNA copies, and you're going to get more number of samples in your hand. With that, you're going to do this experiments. The different types of experiments can be done. This can be even done for therapeutic or pharmaceutical, or to diagnose any diseases, or, as I said, in a crime scene to find out who the culprit is. Or otherwise also, we must have seen some different cases where in the hospitals, the parents fight that the child has got exchanged. So they want to know the paternity test. So paternity test can also be done with the help of this PCR technique. That is, here you're going to amplify the DNA and you're going to do this experiment. How? That's again with the electrophoresis technique. So after the PCR technique, what is that? Now you have got the samples. You want to do the experiment. So how you're going to do this experiment? Here comes the gel electrophoresis technique over there. So what is this gel electrophoresis? You're going to prepare a gel, and in that gel, you're going to add your sample of interest, and you're going to give some current so that the electric current passes through the gel and the proteins move along according to its molecular size over there. So that is how you're going to check the different types of DNA ladders. So once the PCR process completed, the resultant amplified segments are generated, and that can be compared with other nucleotide segments from a known source. What is a known source over here? It can be a known molecular weight, or it can be the standard we are going to use over there to find out whether it is correlating or no. So the PCR generated nucleotide sequence are then placed next to known nucleotide sequence. So this is a known nucleotide sequence, and here you're going to add the unknown samples over there. And then electric current is run through the gel, and the various nucleotide sequence within the gel form bands that resembles a ladder. So you can see this ladder over there according to their electric charge and molecular size. So this is a charge, that is how positive they are or how negative they are. The opposite poles attract, or the like poles retract. So they're going to move away towards the opposite poles over there. And this is termed as gel electrophoresis, and the bands or ladder-like steps that migrate from to the same levels in the gel show identity of the nucleotide base pair. And this method is very popular to complete the PCR test. Now, how is this and what is this? I'll come back to this picture over there, but I want to show you all how this gel electrophoresis looks like. Now, this is what this gel electrophoresis looks like. So you have this gel tank over there. So that's a gel tank over there, and that is the casting tray. You can see, and in the casting tray, we have prepared this gel, agarose gel that is being prepared over there. And you have the small wells that has been done. And into this small wells goes your protein sample of interest, the DNA or the protein of your interest. And they are attached to the cathode and the anode. The opposite ones moves as far as possible. Say, for example, if there are more amount of, say, for example, this cation and anions, they're going to get separated. The positive, the negative charges that are present over here runs towards the positive side. And the positive ones that are present in the samples, it is going to stay near the negative cathode over here. So that is how we are going to get this ladders after the current has been given to them. When they are moving away, when the sample is moving downwards towards the anode side, you can see the proteins getting separated according to its charges and according to its molecular size. That is what we were talking about. So the last one that is moving away is the low molecular weight analysis, because they are very low in size and weight, and they are going to move very faster. Whereas the higher one is going to stay where they are, because they are very heavy and they cannot be run with the help of the charge over there. So this is the hardware that you can see. So this is the gel tank that you can use. This is the power supply where you're going to supply power to this particular gel tank. That's a casting tray. This is all made up of fibers over there. And you're going to put a sellotape on either side, and then you're going to add the gel. Once you add the gel, it is going to take time for it to get solidified. And you're going to add these combs over there. So to cover up all these, you're going to use these combs. And then you're also going to keep a comb-like structures in between. When you have cast the gel, you're going to keep a comb so that it makes the well. Once you remove that comb, you have those wells, and in those wells, you're going to add your samples, and then you're going to pass the current, and then you're going to have this ladder. So this is the ladder that you're going to check after you're running this gel electrophoresis. You might be asked this gel electrophoresis also in your exam. Yeah, you can write this answer if at all they are going to ask what is gel electrophoresis. Now, coming back to this particular pic over there, what we were talking about, this picture over there. So this was a sample that was
Excuse me, ma'am.
Yeah.
Ma'am, in the gel electrophoresis, in the first one, when we are doing elongation, we are creating more and more number of DNA strands, no?
Yeah.
Here we are separating the proteins. What is use of this? I'm not getting that gel electrophoresis.
We are separating the proteins, and what is the use? Right. So now, for example, I was coming to that only. If you understand this picture, you'll understand what is your question again. So what we mean to say is, for example, if I'm going to take this picture of that is who is guilty. Say, for example, there was a crime, and in the crime scene, you have 4 people. Say, for example, you have Bob, Sue, John, and Lisa at that crime scene. and the victim is hospitalized, and or maybe the victim must have been dead. So you want to know who is the person who must have done this crime over there. So there are three innocent person and one is a culprit over there. Okay, so what you're going to do is you're going to take the samples from that crime scene over there, and then what you're going to do is, because the sample is very, very less, you have to undergo that PCR technique, wherein you're going to amplify the DNA, because you have enough DNA now. What you're going to do is you're going to run a gel electrophoresis technique. So what is this gel electrophoresis technique? Now you have taken the blood stain from the victim over there, and you're going to run this gel electrophoresis over there. The victims, from the victim's blood stain, that is, this can be the blood stain of the culprit who must have done that crime. So that is the blood stain from the crime scene. Okay. Now you have four different suspects over there. What you're going to do is you're going to take the blood sample from four different suspects over there. That is from the Bob, Sue, John, and Lisa. This is a general name that they have given. And then what you're going to do is you're going to run a gel electrophoresis. Once you run the gel electrophoresis, this is the bands that you find over there, the different fluorescent bands. And then you're going to compare how, which is the one that is nearest. So for example, this is the crime scene blood stain. This blood will coincide with the culprit over there. Who is the closest culprit? Now, if you can see over there, if this is the first stain, the band over there that you have got, it is coinciding with this first suspect over there. The second band also is coinciding, but the third band is not coinciding. So here again, there are some bands that is running over here, but here again, they are not coinciding. Okay. Then you're going to check the second culprit over there. In the second culprit, there are also very few bands that are coinciding in this particular suspect over there. Then we are going to talk about the third suspect over here. The third suspect, you see this band is this band coinciding. These two are coinciding with this. This is coinciding with this, and these two with this. These two with this, and that with that. So almost all the bands are coinciding with this particular suspect, whereas the last suspect also, not all the bands are coinciding over there. So whom will you say that who is a culprit at this crime scene? You will definitely say that this person is the culprit. So that is the importance of gel electrophoresis. Followed. So here in this suspects, you will have enough samples. You don't have to worry for the samples. But here in the crime scene, you have very less, very less amount of sample over there. So it can be a hair strand or it can be a blood stain over there, which is very less. So what you're going to do is you're going to get very less sample of DNA, and that little sample of DNA is not enough to run the gel electrophoresis. So that is the reason why you're going to amplify the DNA, and then you're going to do this experiment to find out the result. So that is the importance of gel electrophoresis. So is that clear, Shweta?
Yes, ma'am.
Okay. So this is the instrument that you'll be using for gel electrophoresis technique. So what is the advantages? So when you're talking about the advantages of this gel PCR machines over there, this is very quick, simple, and most accurate technique used to create unlimited copies of DNA from just one single strand over there, the one original DNA segment over there. And therefore, the PCR can play an important role in genetic testing and research. And the most common advantage of these reactions are they are highly specific technique that distinguishes DNA sequences by just one nucleotide. Very useful technique when the amount of DNA is very limited, and it has a various applications in the genetic testing and research. Very much useful in forensic laboratories because it is used to investigate the crime scene, and also the paternity test. Here, that is what I was talking about, the paternity test. When there is an exchange in the child, or if the parent says that they have a doubt when the child has been exchanged, what they do is they take the DNA from the child, and then they take the sample from both the parents, that is the both fathers who have this doubt, and then they run the PCR electrophoresis. Then what they see is they can find out the child belongs to which parent. So that's how they clear this doubts and questions in the minds of the parents. And this is very rapid, efficient technique because it can amplify a small amount of DNA to millions of copies. So that was advantage. Now, what is a disadvantage or the limitations for this particular PCR? The limitations of here is sometimes it can be prone to errors and can lead to mutations in the fragment that has been generated. Now, what is the mutations? So, for example, here you can see the PCR. Now, a Taq polymerase is adding the nucleotide base pairs on it, and as it is adding this nucleotide base pairs, sometimes there can be copying errors over there, and that can lead to mutations over there. So that is one problem over there, and they can be altered by non-specific binding of the primers to other similar sequences on the template. Non-specific binding is, we must have heard, there is one more different problems where instead of A, G gets bounded, instead of C, T gets bounded. So that is again non-specific binding that can occur. And in order to design the primers to generate a PCR product, some prior sequence information is usually necessary. That's what I said. The primers should be known primers because the DNA that you're going to separate and want to make copies, that particular site where it has to go and attach to the DNA strand, that should be known. So for that known DNA strand copies, you're going to prepare a primer which is going to be complementary to where they have to attach and grow the length over there. So those are the limitations, but otherwise these are the best technique ever evolved in this decade. So coming to the next one, so that was about PCR. Now we will talk about one more small technique after ELISA PCR. They might ask you for a short answer. Describe dot immunobinding assay. This is a very, very simple assay, very sensitive and very rapid. Dot immunobinding, something like when we are talking about ELISA over there, how you're going to attach those antibodies and antigens. So on that basis, this experiment is done. Dot immunobinding assay. Why? Because you're going to have some dots like things like that over there. We are going to add very, very less amount of samples of antigens or antibodies. So this immuno-dot blot technique is a rapid, sensitive and quantitative method for diagnosis of infections in the fishes in both infected tissue culture and lysates and tissues. Now, why we are going to talk about fishes over here? We are kind of talking about aquatic biotechnology. So here I wanted to keep this fishes over here, because when you're talking about this techniques, we somehow get deviated to the human diagnosis over there. So that is the reason why I again had to add in this fishes over there. So dot plot is also used for diagnosis of infections in the fishes. This can also be used even in the human diagnosis also. Okay, so this, where all you can use this, infected tissue culture lysates and tissue homogenates of the fishes. You can have this samples and you can do this experiment. Uninfected tissue culture lysates and fish homogenates do not react to the peroxidase dot blot system after appropriate treatment. Why they do not react? Because they don't have that antigen to be bound to that particular antibody. As simple as that. So if there is no disease, then definitely you will not get any result over there. If at all the fish is having that disease, definitely you will have a positive result. So the immuno-dot blot method takes 4 hours to complete, and the dot blot membrane can be stored in the dark for a longer period of time. So what is the experiment and the procedure over here, if you can see? So that's the extracted protein. That is, for example, it can be a serum, or it can be a tissue lysates or a tissue homogenate from the infected tissue. So you're going to take the sample, and then you're going to transfer this proteins onto these dots over there. So they have a membrane. So that is a nitrocellulose membrane, the blue color membrane. And they have some spaces where you can add your protein of interest on these dots over there. You're going to add those protein of interest. And then after adding that, you're going to incubate it for a few minutes to hours. And then you're going to add secondary antibodies onto this plate over there. So once you're adding the secondary antibodies, if at all there is the antigen present in that sample, the antibody is definitely going to go and attach onto its antigen. So that is the second step over there. Then in the third step, what you're going to do before going into the third step, you're going to wash the samples. Why we're going to wash it? If there's any unspecific antibodies which have not been bound, they're going to get washed off. And then the third step, you're going to add secondary antibody attached to the enzyme So something like your ELISA, the antibodies has been attached to an enzyme over there. That is a secondary antibody. And once you add the secondary antibody, if they are specific, they are going to get attached onto this primary antibody over there. And then this is going to be incubated, washed. Why washed? Again, because if there's any extra secondary antibody, they are going to be washed off. Then you're going to add a substrate. What is a substrate over there? The substrate is going to get attached onto this enzyme over there, something like this. A substrate is a colored product over there. And this is going to get attached over there to give that particular color over there. So that is how you're going to do this particular dot plot technique. So how it is done, now if you can see in this the second picture over there, so these are carried out with 2 antibodies and 1 antigen. What is antigen over here? This is the fish lysate homogenate tissue homogenates or lysates over there. So it is going to be bound to the antigen with the help of 2 antibodies over there. The provided strip has 3 pads of nitrocellulose membrane. So it is going to have these 3 strips of nitrocellulose membrane. The lowest pad is going to be for the positive control sample. Positive control is they are going to have definitely an antigen and the antibody is going to get attached to it. That is positive control. The middle one is a test samples where you're going to test the patient sample or fish sample over there. So that is the test sample. And the upper one is a negative sample, wherein it is not going to have any antibodies present in them, antigens present in them. That is a negative control. So these strips are used to find out the presence of antigen in the given sample. So how are they done? The strips are incubated with a test sample containing antigen. So that is, you're going to add your sample. Then you're going to add your primary and secondary antibodies, that is HRB conjugated secondary antibodies. And the addition of the substrate, so DAB is a substrate that is going to be used, which is going to give you all the brown colored spot over there. And if the test sample does not contain an antigen specific to the antibody, there will be no enzymatic reaction and no spot will be developed. So what is the interpretation that you get over here? The presence of the spot on the positive control confirms the proper performance of the reaction. So that means you have carried out the proper performance of the reaction, which shows that everything went on correctly. The spot observed in the middle pad indicates that the presence of antigen in the test sample against the antibody. So that means the patient is positive to that particular disease is what you are going to confirm. Now, if you can see in this second picture, there is no dot present over here. So that means the patient is not having that disease. He is negative to that particular disease is what you're going to conclude. And the absence of a spot in the negative control ensures no cross-contamination has occurred. So that is, the process of experiment has been done correctly. That is what we are going to talk about. So that is what is dot immunoblotting technique over here, or dot ELISA. It's a very, very simple technique, and it is going to get finished within 4 hours. And this is what is being talked about in this particular dot ELISA technique. So this, they might ask you all for a very short question, that is for a short answers. They might be asking this particular question over there. Okay, so that was about immunoblotting. So we talked about ELISA, we talked about PCR, and we talked about dot plot. So aquatic biotechnology, we talked about how you can do these experiments for the aquatic purpose. Now, when you're talking about aquatic biotechnology, how can we forget about pearl production? Because pearls are the females, again, on par with diamonds. Pearls have also been with us, and of course, Hyderabad are pearl lovers from the Nizam era. And we were the first ones to have a lot of Hyderabadis, or the Nizams were the first ones to love this pearls. And though we were producing good enough pearls of late, for the past 2 or 3 20 or 30 years back, somehow India became backward in production of pearl, and we are now very much far behind when you compare what is the world going to produce. The first one, of course, definitely China, because that is where the pearl production started, China, then Japan. Japan concentrates on marine pearl culture, whereas China concentrates more on freshwater pearl culture. So we are going to talk about two types of pearl culture. One is a marine pearl culture and one is the freshwater pearl cultures. And when you're talking about marine, then definitely you're going to talk about these oysters. These oysters are going to be found only in the marine, and the other mussels, when you're talking about the mussels, we're going to talk about freshwater culture. Okay, so biological approach of pearl production. This is one more small topic that has been discussed in your textbook. So I just thought let's complete this point also. So the pearl oyster belongs to the genus Pinctada, family Pteridae, the class bivalve, because bivalve is those two valves that you're going to have. So that's a bivalve, and the phylum Mollusca. The important species producing good quality of pearl is Pinctada vulgaris. So here we are going to talk about the marine pearl production over there. So when you're talking about the marine production, Pinctada vulgaris. So this is this is Fucata over there, and that is vulgaris over there. The name that has been given, and the second one is Pinctada margaritifera. So this is margaritifera. You can see those sharp edges that it's going to complete. So that is margaritifera. Then Pinctada chemnitzi and Anamoides. Then you also have Atropurpurae. So mostly commonly found in India is Pinctada vulgaris, and but mostly this Fucata is also used for culturing the marine pearls over there. So here, when you're going to talk about this Indian pearl oyster Pinctada fucata over there, is used for production of cultured pearls. Cultured is you're going to culture them. This is not natural. You're going to culture. You're going to interfere with the nature, and you want to culture this particular pearls over there. So that's called as pearl culture. And this is production technology was perfected, and the technique involved was standardized. Manipulation of the color and the quality of cultured pearls was rather difficult in vivo culture, and hence an attempt to in vitro pearl production was made through this tissue culture technique. So what is this? Now, if you can cut open, so this is the mantle you can find over there, and inside the mantle you have this pearls over there. And if you're going to check into what is the different parts of this oyster over there, so you have the shell, that the outer covering is a shell, and that is the mother of pearl, that is the nacre, where you're going to have the pearl of that color, whichever the color the nacre is, that color is where you're going to have that particular pearl colors. Then that's a mantle over there. So that's a flesh where it's going to start. So that is called as a mantle. Then you have the anus of the oyster over there, the anus. That's the adductor muscle. And it's going to attach the oyster tightly. So that's the adductor muscle. Then they have those gills over there. So that is the gills. Then a pea crab. What's a pea crab? The crab must have entered inside the oyster, and they start living inside. That is not the part of the oyster anyways. That's the foot over there, then the mouth, the stomach present over there, and also the gonads that is being present. So these are the different parts of the oyster that we'll be talking about. Yeah, I not able to listen. Are you able to listen now? Am I
Yes, ma'am, we
audible?
are able to listen. Yeah,
Yeah.
yeah, it's audible.
Okay. I think Annapurna has said that she's not able to listen. Am I audible, Annapurna? Because others are able to listen to me, so I don't think so there's any problem. Okay, so that's a mouth over there, then the stomach and a hinge where it is going to where you can just open it like that. That's the hinge over there. So that was the different parts. So what is production of the pearls over there? So you have two types of production. One is the natural way and one is the artificial way. The artificial way is the technology that we are going to talk about in this particular class. In the natural way, you can see the natural pearls are formed due to the natural processes or disturbance in the environment where the oyster lives. And when you're going to talk about the natural pearl, this is the layer, the concentric layers that you can find over there is what is a natural pearl. And this concentric layer is the conchiolin and the nacre deposition that is going to be argorite deposition. And this is what is a natural pearls, which is going to be very, very costly in the market. It can go up to some thousands of dollars if you're going to have this natural pearl. Now, this is a cultured pearl over there. What's the difference between a natural and a cultured pearl? The natural one, when any foreign particle goes inside, any tiny particle goes inside, the tiny particle is covered by this nacre or the mother of pearl completely. But when you're going to talk about the cultured pearl, this particular blank area over there is that large size of nucleus that you're going to introduce, the artificial beads or something like that, you're going to introduce into the mantle of the oyster, and then the oyster is going to add on its nacre over there, that is the mother of pearl, the shine. It's called as a nacre, that is a protein part of the oyster, which is named as nacre, N-A-C-R-E, nacre. Now that is going to be produced around this pearl of nucleus. Pearl nucleus is the artificial pearl that you have prepared, and then you're going to introduce it into the oyster, and then after some days you're going to see that it has produced that nacre around them. So that is a cultured pearl, and that's the reason why we are able to afford pearls, because they are cultured and they are a little bit less costly than when you're talking about the natural pearls over there. So artificially inducing the mollusca to secrete the nacreous matter around the inserted nucleus, and it is produced by human interference and is known as cultured pearl. And the chief imitations made of plastics that have an artificial lusters are known as artificial pearl. So you can see how the natural one has that beautiful color over there and how a cultured pearl is differentiated. The cultured pearl will not have that much of color as a natural pearl has. So natural pearl radiates whenever the sunlight falls on it, they just show that shine over there. So here you're going to talk about how these are being prepared. So natural pearls are formed when an irritant like a parasite, a piece of a shell or a snail finds its way into the mollusk, rarely a piece of sand, and lodges between the shell and the mantle. So if you can see in this picture over there, so that's the mother of pearl nacre layer. And if a foreign body enters inside, it goes through inside the mantle area. Once inside the mantle, what it does is the epithelium, the mantle epithelium is going to cover over them, is going to make a cover around. And as it is going to be covered, the shell, that is the mother of pearl nacre layer, is going to produce that proteins around them. Why they're going to produce that protein is, what it is doing is something like macrophages. Remember a macrophage? The macrophage is going to engulf the parasite and they're going to form a cover around them, and then they're going to digest. So in the same way, the mollusca or the oyster, what it is going to do is they're going to produce its protein. Now this protein is a foreign protein for that particular parasite. So that means the antibody is going to be produced around them and they're going to engulf. So in the same way, the mother of pearl, the nacre is, that is the protein, is going to be covering the parasite completely around, and that is what you can see over here. So that's a concentric ring that has been found, and then after it has been produced enough proteins around them, they are going to be separated over there. So that is how a formation of pearl occurs in a natural way. So when an irritant becomes trapped in the mollusca, the animal senses the object and coats it with a layer of aragonite and conchiolin. So this is what is we are talking about, the proteins of the nacre. So the nacre is made up of aragonite and conchiolin, and this aragonite and conchiolin are going to be something like this layers. So they are going to be an hexagonal type of layer, arranged one above the other, something like a brick arrangement. They're going to be arranged one above the other, and that is going to form a complete round around the foreign body over there. So that's a six-sided crystals that is going to be formed. Which one? The aragonite is going to be arranged as a six-sided crystals, and between each sheet, the mollusca is going to secrete a very thin layer of membrane forming the protein conchiolin. So here you can see, so that's the nacre layer. You have a conchiolin layer and you have a aragonite layer over there. So this the parallel formations of an aragonite and a conchiolin layer, aragonite layer, conchiolin layer. So that is going to be forming a round one like this. So this is what you're going to see if it is going to be inside. And that's a bead nucleus. That is when you're going to talk about this culture. So that's a bead nucleus, or it can, if it is a natural one, this can be a any foreign body that has entered into the mollusca, so that the nacre is going to be produced on them. Okay, so how this is being done over here? So this is the exterior shell of that margarita. Can you see those sharp edges of this? So you can take it as a margarita over there. And then this is the prismatic layer that is formed over there. That's a prismatic layer, that the shell interior. So this is the nacreous layer. This is the part that is going to be produced around the pearl on them, and that's a mantle. If it goes inside the mantle, within the mantle, the shell that is going to be present underneath this shell interior, that is going to produce a nacre around them, and that is when you're going to get that particular luster. So whatever this luster is, that luster you find it on the pearl. Okay, so the basic step involved. So what is the basic step involved? The freshwater pearl culture. So here we are going to talk about a freshwater pearl culture. We've completed the marine water cultures. We're going to talk about the freshwaters. So when you're going to talk about the freshwaters, there are three important mussels that you're going to talk about. That is Lamellidens marginalis, Lamellidens corianus, and Peresia corrugata. So this is marginalis, this is the corianus, and that is the corrugata. So the different types of mussels that you're going to talk about in the culture of freshwater pearls. So the summary in the steps of yours, first of all, you're going to collect the mussels. These mussels are collected at the Tungabhadra and different type of places where you can find those freshwater mussels growing in them. So you're going to first collect them. Then you're going to do a pre-operative conditionings. That is, you're going to allow them to get ready for the operation. And then you're going to implant the graft inside them, and the post-operative care is taken, and then implanted in the mussel. That is, a pond culture is done. And then you're going to harvest it after, say, around 8 months or 11 months, possibly, depending on what is the size you have infused. What are those steps over there? I'll just quickly go into the steps over there. So first thing is, you're going to collect the mussels. So you're going to collect it from any river beds over there. First, you're going to do the collections over there. You have to find out which type you're collecting, because you should know how does a marginalis looks like, or a corianus or a corrugata looks like. So you have to be very sure. An expert has to be there to collect this particular mussels, because other mussels are of no use to produce this particular pearls. So ideal sizes 8 centimeters anterior to posterior. So that is the anterior to posterior. You're going to have an 8 centimeters. A medium size is very much important, because if you're going to go for a larger ones, the larger ones are already aged, and when you're going to do this operation, they're going to die in this process. So the best one is a young ones, that is at 8 centimeter possibly. And pre-operative conditions. So what you're going to do after collecting, you're going to condition them for 2 to 3 days by keeping them in a crowded condition. So you're going to have a cement tank, and you're going to collect all those mussels, and you're going to keep it in that crowded condition. What is this crowded condition? 1 mussel per liter of water. So 1 mussel should have 1 liter water. You're going to keep, that is, you're not going to keep one in a bottle. You're going to construct a pond like that, and then you're going to leave all the mussels in that particular pond. So that is at 1 mussels per liter of water is what you're going to talk about. Now, what is this preconditioning operation is about? You are allowing them to weaken the adductor muscles. So to open a muscle, it is going to be very, very tough. So when you're going to keep it in a crowded condition, because of that congestion, they want to breathe in. So then they slowly loosen up the adductor also. They slowly loosen up the adductor. So that is how you're going to, that is the reason why you're going to leave them in a crowded conditions over there. And then muzzle surgery. So what is a muzzle surgery? So you're going to clean up all this mantle, that is exterior skin over there. You're going to clean up, and you're going to try to open it up. So why you want to open it up? Depending on the place of the surgery, the implantation is of 3 types. You can either keep it in the cavity, you can either keep it in the mantle tissue, or you can also leave it in the gonadal implantation. Gonadal implantation. So wherever the gonads are present in the muscle, you're going to leave that implantation in the gonadal region. So 3 regions: cavity, tissue, and gonadal implantation. So the key material required is the surgical implantations. For that, you need either a bead or a nuclei, which are made usually by the molluscan shell. If it is a bead, you can make it by the molluscan shell or a calcareous material. If it is a nuclei, you can take the mantle itself, the donor mantle itself. What is this donor mantle? Now, you're going to talk about first the mantle cavity implantation. So this is the cavity implantations that we'll talk about, the first one over there. So in this procedure, around 4 to 6 mm diameter or a designed bead, can you see the bead? This are designed bead over there. These are inserted into the mantle cavity region of the muzzle after opening the two valves. So you're going to open this two valve with the help of a forceps and a stick over there. So once you are going to open it up, you're going to introduce, so this is a rod wherein you're going to take a designed bead, that's the artificial one, and you're going to insert it into the muzzle's cavity over there. So you're going to leave it into the cavity, and then without causing any injury, you're going to leave it into the cavity. And then what you're going to do is, after inserting it inside over there, then you're going to close the valve, and then you're going to leave it in the post-operative tank over there. So that is one type, that is a mantle cavity implantation. The next one is a mantle tissue implantation. So mantle tissue itself, you're going to add over there. So what is that? You need a donor and a recipient over there. So when it's in a donor, the donor can be a old aging muzzle over there. So what you're going to do is, you're going to cut this mantle, and you're going to take a long strip of this mantle over there, and you're going to cut it into very, very tiny ones, that is 2 into 2 mm is what you're going to cut them into these pieces over there. Now this becomes the nuclei, and this is what you're going to insert into the recipient muzzle over there. So a donor muzzle and a recipient over there, that is a grafting. So what is happening over here? The donor and the recipient muzzle is to be taken. The first step in this procedure is to preparation of the graft. So we have done this graft, and the implantation is done on the recipient muzzle, which is of two types, non-nucleated and nucleated. So in a non-nucleated type, only the graft pieces are introduced into the pockets of the posterior pallial mantle present at the ventral region of the muzzle. And in a nucleated method, a graft piece followed by a small nucleus 2 mm diameter is introduced into the pocket. So what it means to see over here, in a non-nucleated, you're going to only introduce the small mantle tissue. So that is non-nucleated type. So when you're talking about the nucleated type, along with this graft, you're going to add even a nucleus. What is this nucleus over there? It's an artificial bead. So both of this goes inside. So that is a nucleated method. In a non-nucleated method, only a graft is sent inside. So that is a difference you have to understand. If at all you come across this bead is, this pearl is made up of non-nucleated type, this bead is made up of nucleated and beaded type, then you have to understand what is the difference between non-nucleated and nucleated. In the nucleated, the nucleus along with the bead is both is inserted, that is graft is done inside this recipient over here. So that is the procedure over there. So you're going to open it up, and you're going to slowly graft these two into the mantle cavity of the recipient over there. So then what happens after this has been added into the pearls pouch? You can see over there. So that is the bead over there, the nucleus over there. That is the mantle tissue that you have taken from the donor over there. And when you're introducing it, the epithelium of the recipient is going to slowly form around them. So you can see they're slowly forming around them. And once they are forming around them, they form a complete structure around them. And from there onwards, this epithelium is going to release the protein. What is a protein? The nacre is a protein that is going to be produced around them, and completely you're going to see the color change over there. So that is the nucleus, the artificial nucleus that you have added. And then after say around 15 months after depositing this nucleus, after 15 months, you're going to see a complete pearl harvest that is being done. So this is a mantle tissue implantation. Under mantle tissue implantation, you're going to talk about non-nucleated type and nucleated type. Again, a non-nucleated type, you're going to send only the graft. This is the mantle tissue that you're going to add. And in a nucleated type, you're going to add both the graft and the nucleus inside them. So that is a nucleated type. Then the last one is a gonadal implantation. So if you can check on this particular oyster over here, so you can see this part is the gonad, the orange color part is the gonad that you're going to talk about, the mantle, then the different organs that you can find over there. And this mostly you can see in the Tahiti beach over there, or Tahiti Sea. So the beaded cultured pearls are gonad grown. So what you can do is over here, so can you see the gonads over there? You're going to make a very small slit near the gonad, and then you're going to introduce the nucleus inside them. And once it is going inside the nucleus, then you can find that particular pearl culture that is being done over there. So this is how, and in this gonadal implantation, you can reuse them again and again. But whereas when you're going to talk about the tissue mantle, they're going to get attached onto the shell, and then you'll have to break it open and separate them. There you cannot reuse those particular oysters over there. In that you can have different shapes of whatever. Nowadays you must have seen the idols, something like Lord Venkateshwara or Sai Baba, or a heart shape or a bird. So whatever shape you want to prepare that particular nucleus, and then once you insert them, that is how they are able to produce the faces of the deities, the god and the goddesses deity, and that we are going to prepare them as a small type of whatever, you can wear it on the chain, so the taller shapes, and you can wear it onto the chain. So that is how you're going to prepare them. So in the gonadal implantation, this procedure involves preparation of the graft as described earlier. We talked about the mantle tissue. First, a cut is made at the edge of the gonad muscle. So a very small cut is made at the gonadal muscle, and then the graft is inserted into the gonad, followed by a nucleus of 2 or 4 mm diameter, and then this is allowed to close back, and then allowed to culture, and then after some say around 15 months, or it can also go up to 2 or 3 years, this can be left out, and then you can collect them, harvest those nucleus. So that is the nucleus that is being harvested after being grafted. So care is taken so that the nucleus touches the outer epithelial layer of the graft, and the intestine is not cut during the surgery. So you have to be very sure the nucleus touches the outer epithelial layer. Why? Because the outer epithelial layer is the one that is going to produce that particular protein, the nacre. It has to produce around that particular nucleus. Otherwise, if it is not going to attach onto it, then the pearl culture is not being done. And when you're doing this experiment, you have to be very sure that you're not going to cut the intestine. If you're going to cut the intestine, then there's no digestion, and definitely the oyster is going to die. So after post-operation, that is after introducing all this nucleus, what is the post-operative care that you're going to take? So implanted muscles are kept in the post-operative care unit in a nylon bags for 10 days. So these are the nylon bags that you can find. This is also one type of nylon bag. You're going to introduce all these oysters after post-operation. That is, after you have introduced that nucleus, you're going to take care. Why you're going to take care for at least 10 days with the antibiotic treatment is because you have done a operation over there. Now the pearl is on the oyster is under shock. What has happened to me? And then you have introduced use some different forceps or sticks, or the nucleus that you have introduced inside. All this can cause any infections into the muscles over there. So that's the reason why for 10 days you're going to keep it under your vision by treating them with antibiotic treatment. So you're going to add some antibiotic medicines in this particular place where they are going to survive, and you're going to see for whether they have accepted the nucleus. One important method of post-operative care is you want to make sure that oyster has taken that nucleus. Sometimes what happens is the nucleus is going to be thrown out by the oyster. So all your work is gone waste, because you are not going to invest your time leaving that oyster in the water for 15 months, because it's not 1 day or 2 day or 1 month or 2 months that you're going to get the result. 15 months, or say 2 years or 3 years. So before introducing these into that pond, or wherever you're going to preserve them, you're going to check it for 10 days, see whether they have accepted the nucleus. If they have accepted, then they are within the body. If they haven't accepted, they are going to throw it out. You will come to know whether it has thrown it out. And sometimes due to this operation technique, sometimes due to bad handling, the oysters or the mussels can die. So all those dead ones can also be removed out. So that is the importance of a post-operative care that you're going to do in this particular experiment. So this is how you're going to keep a 2 to 3 meter nylon bag, and you're going to keep those mussels over there. And this is also a way you're going to take care of them. Okay, so harvesting. So after 15 months or 2 years or 3 years, definitely you want to see what is the result that you have done. So this takes between 6 months to 4 years to develop the pearl. And during this time, the pearl farmers take care of the oysters by feeding them. What is the feed that they are going to be given? Because these are filter feeders, they are going to filter the water. They take in the water, filter the water, and what are the micro organisms that are going inside, they are eating it them. So phytoplanktons, mostly algae. So what some people do is, when I had come across this culturing, what they do is they even add some groundnut powder along with the algae cultures. So they're going to mix this culture, and they're just going to spray on the place where they are going to survive. And this small tiny micro particles, they're going to go in, and you're going to allow the oysters to survive. So that is one way of taking care of the oysters during this 6 months to 4 years period. So and by that feeding, along with that feeding, you're also going to check for the parasites, and you're going to ensure that the water conditions are optimal for the development of the pearls. So when the pearls are ready to be harvested, pearl farmers remove the oyster from the water, and harvesters open the oysters, remove the pearls, and in some cases prepare the oysters to create more pearls. So prepare the oysters to create more pearls. Over here I was talking about the gonadal technique. So this is the gonadal technique over there, because once it goes inside, the gonad is going to get healed up, and after removing it, also again you're going to make them to get ready for the next operations over there. So that is one way of reusing the oysters. And sometimes when you're going to cut open the shell and remove that idols that you have prepared, then you cannot reuse those oysters or mussels over there. And this is an extremely delicate process, and one requires gentle handling to ensure the pearls, including the rare and the high value pearls of Australia, Japan, Philippines, and other countries, and they are not damaged in the process. Because when you're removing it, the pearls, you're going to be very careful when you're removing it out, and you're going to take care of them, and you're going to harvest them, because a complete round without any up and downs is going to fetch you all thousands of dollars. That's the reason why they are these pearl farmers are going to be very careful. And this pearl farming, anyone can take it up, because they don't need larger land like cultivation of paddy over there. And also you need to have very less place, and you need to have a lot of patience. And if you have all these things, you can start off your own pearl culturing, because government is also giving a lot of fundings to for this pearl farmers, because India is very much far behind when you're going to talk about international pearl cultivations, because it's going to give us some billions of money over here. Definitely government is definitely giving some support for pearl farmers over there. You can have 3 day or 4 day courses over there. Bangalore is one where you have this government run place which gives you all this pearl culture farmer techniques. So you can just go enroll yourself, and then you can learn how to culture them. And definitely you people can also become pearl farmers by gaining good interest in this and culturing them. So this is how a salt water and a pearl, a freshwater pearl looks like. Can you see the luster in the salt water? Look at the shine that is been given in the salt water, and then compare it with the fresh water. Why? Because salt water, they take longer time for the formation of the pearl, and it's going to have a very high quality luster, and definitely they are very very expensive ones. So you can culture them even in the salt waters. And the fresh waters, these are sometimes they are natural, and sometimes they are cultured. And of course they take very less time. 15 months is enough for them to culture, whereas for the saltwater pearl cultures, you need at least 3 to 4 years minimum for culturing them. But when you get the result, then definitely you are going to earn in good amount, some in dollars, you can say. And so this is how you're going to check for this pearl that has been formed over there. So that was about the pearl cultures and aquatic biotechnology. So So with that, we come to the end of this topic. Any doubts, Ma? Any doubt in this topic? So, okay, so if you are not having any doubt, so we'll wind up this session. Meet you on in the next session tomorrow. Okay, see you all.
Tomorrow is there, ma'am?
Yeah, we do have class. Why? Any doubt on that?
Yes, ma'am.
We
No,
do.
no.
Yeah, we do
Yes,
have the
ma'am. Thank you, ma'am.
So tomorrow will be
Yes,
the
ma'am.
last class because we have completed all the 10 days classes. Okay. So tomorrow
So
will be
tomorrow is the last day, ma'am?
yeah, tomorrow is the last class.
Okay.
Yeah,
Okay,
and
ma'am. Thank you.
yeah, I'm available anyways for you all on the WhatsApp. So any doubts, you can get back to me on the WhatsApp, right?
Okay, ma'am. Thank you.
Okay, ma'am. Thank you.
Yeah, thank you.
Okay.